Post-fertilization structure and events. The events and the changes that take place in the floral parts of a plant after fertilization are the endosperm development, embryo development, and the formation of the seed, consequently the fruit. The parts of a flower transforms itself after fertilization. The sepals, petals, stamens, and stigma they usually wither and fall off after fertilization. The ovary gives rise to a fruit, the ovule gives rise to a seed, and the egg that forms a zygote. The zygote after division it gives rise to the embryo. This is fed by the nutritive tissue endosperm. Endosperm is formed from the secondary nucleus of the ovule as we have learned earlier. So some of the parts are before fertilization and after fertilization in a flower. Post-fertilization event one is the endosperm development. Based on the mode of the development, endosperm it is divided into four types. Nuclear endosperm, so this is the zygote and this is the primary endosperm nucleum. It starts dividing before the zygote starts to divide because it has to feed the zygote. So if it divides into two without formation of any cell wall, it gives rise to a free nuclei inside, and this type is called as a nuclear endosperm. If the primary endosperm divides and after two divisions a cell wall is formed as we are seeing here, this will form each nucleus is divided into cell wall. This will be called as a cellular endosperm. For this we are seeing an example of the coconut. Coconut it is having both liquid endosperm and the solid endosperm. Solid endosperm is formed from cellular endosperm as example of cellular endosperm. Liquid endosperm is an example for nuclear endosperm. Helobial endosperm, here the nucleus it moves towards the chalazal end. This is the micropylar end and divides into two. Near the micropylar chamber, it divides without the cell wall formation in a larger space, and chalazal end it moves towards the corner of the chalazal region, and here it divides with the formation of the cell wall. This type of development is called as a helobial endosperm, and as we see in hydrilla. Ruminate endosperm. In this endosperm, this is an example of Areca catechu park. Here, what happens is that it's an irregular surface of the endosperm that we see in ruminate endosperm. The development of an embryo is another post-fertilization event. Here, we are seeing the development of the dicot embryo. The first formed cell after fertilization of the ovule and with the anther is forms the zygote. It divides into two and then it divides further growth, and this embryo this will be called as a proembryo that I am showing here, and this proembryo undergoes several divisions to form globular embryo, heart-shaped embryo. Finally it becomes mature embryo. Before coming to mature embryo it undergoes different stages: globular, heart-shaped embryo. The mature embryo has plumule, cotyledons and radicals. These are the embryonic. Radical is the embryonic shoot, cotyledon is embryonic leaf, plumule is embryonic shoot. Here. Then the next structure that we are going to see after the fertilization event is the seed. Seed, dicot and monocots. The difference only is that in the cotyledons. The seed has an outer covering called as the testa and the inner covering tegmen. From the radical only it is the embryonic that is we are seeing here. This will be the embryonic root. Cotyledons are the embryonic leaves, and as we have seen here, plumule is the embryonic shoot. These cotyledons, basically if there are two cotyledons, it will be a dicot root. If there is only one cotyledon, as we are seeing in oral sativa here, this is the endosperm. This is going to feed the growing embryo. This embryo is having a scutellum. This scutellum absorbs the nutrition and provides to the growing embryo. Shoot apex, it is covered by a sheath called as a coleoptile, and here is a root cap. Root cap beneath it is radical, and the root cap above the root cap is coleorhiza. That is the sheath which is preventing or which is protecting the radicle. We know fertilization involves the union of male and female gametes in plant. If reproduction or the embryo develops without the union of male and female gamete, the process is called as apomixis. It was first introduced by Winkler. The term was first introduced by Winkler in the year 1908. Maheshwari, he classified this apomixis into two types, recurrent and non-recurrent type. If the haploid embryo develops, sac develops into a embryo. That is, after meiosis, we know 4 haploid megaspores are formed. Of these 4 haploid megaspores then undergo fertilization. That is a normal process. If the haploid megaspore develops into an embryo sac without fertilization, it will be called as non-recurrent apomixis. In recurrent apomixis, as we are seeing here, it is classified into 2 types. Vegetative reproduction, that is, any part of the plant, if it propagates into a new part other than the seeds, without the involvement of the seeds, it will be called as vegetative reproduction. And then agamospermy, in this process, embryos are formed, eliminating meiosis. Totally meiosis and syngamy is not found, which is a normal process found in fertilization. How embryo sac will develop then? In adventive embryony, if any cell of the nucellus or integument, it gives rise to an embryo sac, then it will be called as an adventive embryony, which is found in citrus. In Diplosparry, what happens is that megaspore mother cell, it undergoes, it doesn't undergo meiotic division. Without any meiotic division, if it develops an embryo sac, it will be called as Diplosparry. This example is Eupatorium. Apospory, if a megaspore mother cell undergoes meiosis but develops into an embryo sac without syngamy, that is, a nuclear cell, the megaspore mother cell divides meiotically into 4 megaspores, but they gradually disappear. And so, a nuclear cell, it becomes active, and if it gives rise to an embryo sac, that process is called as apospory. Example for this is Parthenium. In polyembryony, more than one embryo is formed in a seed. The process is called as polyembryony. It was first reported by Anton van Leeuwenhoek. The picture we are seeing here, see, zygotic embryo. A single embryo is the usual nature. If it cleaves and forms many embryos, and here antipodal cells, if it gives rise to a embryo, that means more than one embryo, if it is found in a seed, the term is called as polyembryony. These are the other processes that are seen in plants apart from fertilization or the formation of a seed, normal seed in plants. Seedless grapes, papaya, banana, all development without true seeds. We know that means seedless means that these fruits don't have any true seeds. How are these developed? These are developed by a very interesting process called as parthenocarpy. The ovary, we know that the ovary becomes a fruit and the ovule becomes a seed after fertilization. But in a number of cases, if the fruit develops without the act of fertilization, that fruit will be called as a parthenocarpic fruit. We are seeing here an example of custard apple, normal custard apple with seeds, and these are seedless fruits. This fruit is developed by a process called as parthenocarpy. So these are many variations in the plants apart from sexual reproduction, normal sexual reproduction, various processes like apomixis, polyembryony, and parthenocarpy take place in plants. So, summary in nutshell, we can say that a flower gives rise to a flower pot in the reproductive part, anther and pistil, the formation of the male gametophyte, the formation of the female gametophyte, and then falling of the pollen grains on the stigma, that is here the fertilization takes place, development of the embryo sac and the embryo, and then from the embryo sac develops in the embryo sac the egg gives rise to a zygote, and then endosperm is a nutritive tissue for the zygote growing a zygote, and these then develop into a seed and then embryo seed coat having testa and tegmen and the embryo. The embryo, this seed develops into a new, germinates into a new plantlet, and then this is the process of the growth in angiosperms. In total, the nutshell, the sexual reproduction seen in plants.